The objective of any chromatographic analysis is simple: capture analytical reality without distortion. We must translate the physical chemistry into an accurate digital measurement. To guarantee that accuracy, laboratories deploy an HPLC integration master. This is a rigidly validated framework, a locked software configuration that dictates exactly how a system interprets raw data. Instead of relying on a human analyst's judgment, the integration master mathematically defines peak width, sets strict rules for baseline correction, and establishes the exact detection thresholds required to identify a compound. Its primary mechanism of action is the algorithmic elimination of human bias. By forcing the software to apply universal rules to every single run, the integration master separates objective chemical reality from subjective human interpretation. Deploying this framework inside a pharmaceutical laboratory achieves 4 specific operational objectives. Looking at this guidance document, the first objective is to standardize parameters across every HPLC system. Second, it physically prevents manual data manipulation. Third, it maintains strict audit trails with permanent timestamps. Finally, it ensures uniform interpretation across all analysts and instruments. By satisfying these 4 objectives, data integrity is permanently baked into the analytical process. The laboratory relies on a verifiable algorithm rather than trusting human honesty. To understand how the system eliminates bias, we have to look at the specific parameter settings that dictate how the software sees a chemical compound. This chart outlines core parameters, starting with the peak detection threshold, the absolute minimum signal required to recognize a peak. Peak width establishes expected shape, preventing mischaracterization. Finally, baseline correction adjusts for systemic drift. Then we have the mathematical integration algorithms, which define exactly how the software calculates the area under the curve. 2 primary methods handle overlapping compounds: tangent skim and valley to valley. A tangent skim draws a diagonal baseline under a smaller peak riding on the tail of a larger one. Contrast that with a valley-to-valley integration, which drops a hard vertical line down to the baseline at the exact lowest point between 2 overlapping peaks. To clean up the data before calculation, the system uses Savitzky-Golay filtering. This is the standard mechanism for noise and smoothing. It mathematically reduces random, high-frequency signal fluctuations without artificially inflating or deflating the total peak area. For highly complex chromatograms, predefined peak splitting rules take over, dictating precisely when a jagged peak is treated as 2 overlapping compounds based on strict resolution criteria. Because these mechanical parameters are established in advance, the software algorithm makes the final decision. This effectively blocks any manual manipulation at the bench. Algorithmic parameters are meaningless if they can be quietly altered by a chemist looking for a passing result. Returning to the guidance, all parameter frameworks undergo formal method validation. Once locked, any parameter change requires strict change control and quality assurance approval. Finally, system suitability tests confirm these parameters apply consistently to known standard samples. The integration master's architecture is permanently locked down. If an analyst deviates from the approved parameters, they generate an unavoidable digital footprint. Algorithms expect perfect symmetry, but physical chromatography is messy. Chemical compounds naturally exhibit fronting curves or tailing factors, smearing and stretching inside the column. As long as these shapes fall within the validated acceptance criteria, they are analytically valid. This creates a common friction point, leading to a major audit failure: analysts manually forcing compliance by improperly chopping off a peak's tail. The incorrect practice is artificially lifting the baseline vertically, halfway up the tail, to exclude the extended edge. This definitively constitutes data falsification, excluding valid peak area that physically passed the detector, artificially lowering reported concentration. If a tailing peak absolutely requires manual integration due to a system anomaly, there is only one strictly approved corrective action. The analyst must manipulate the start and end time selections horizontally along the true natural baseline, rather than altering vertical height. You define when the peak starts and stops, keeping the baseline at the floor. The master guidance outlines legitimate handling for anomalies. For baseline drift, re-equilibrate the column. For peak splitting, optimize approved parameters. For negative peaks, check physical detector settings. Ultimately, manual reintegration must be heavily restricted and permanently logged. Manual intervention is sometimes an analytical necessity, But under the integration master, it can never be used as a shortcut to pass a failing batch. Shifting our perspective from the laboratory bench, we face the strict expectations of global regulators. These algorithmic constraints are governed by international pharmaceutical law. Looking at the regulatory references, ICH Q2R2 acts as the governing mandate for formally validating these analytical procedures and proving the math works. FDA 21 CFR Part 11 enforces the strict governance of electronic records, ensuring the audit trails capturing these parameters cannot be deleted. European Union Annex 11 demands similar rigor, dictating the validation requirements for all computerized laboratory systems. WHO TRS 986 stands as the ultimate global standard for complete data integrity in analytical laboratories. Circumventing the integration master constitutes a documented breach of international regulatory frameworks. Ultimately, the HPLC integration master serves as the absolute final barrier between the raw, messy data and the official final report. Its purpose is to ensure that the final reported results are a flawless, standardized reflection of physical chemistry. Human bias, whether intentional or accidental, is the single greatest threat to analytical truth. Implementing a rigid, validated integration master is the only way to definitively safeguard GNT data integrity from sample injection to final release. To deepen your understanding of laboratory compliance and master analytical quality control, subscribe to the Pharma Training channel for more cinematic GMP and technical learning.